What if I told you that some of the most powerful cancer-fighting compounds ever studied by modern science aren't inside a hospital or inside a prescription bottle, but sitting in your refrigerator, your spice rack, and the produce aisle of your local grocery store? Most people have no idea. We're spending billions every year on treatments that work on the tumor after it already exists, while the foods that help your body prevent those cells from ever going rogue in the first place are being ignored, overlooked, or left to rot in the back of the crisper drawer. Today, I'm going to show you 20 of them. Foods backed by genuine peer-reviewed research, studied in universities and cancer institutes across the world, and eaten by the longest-lived populations in human history. Stick around, because number 3 on this list is a purple vegetable so packed with anti-cancer compounds that researchers are now studying its pigments as a potential template for new therapies. And number 12 is a berry so consistently studied in cancer research that it has its own dedicated scientific literature almost nobody in mainstream nutrition talks about. And if you stay until number 20, I'll show you something from the ocean that contains a compound so unique no land plant on Earth produces it, and cancer researchers have been studying it for decades. Make sure you hit that subscribe button, because every week we bring you the deep science behind the foods your body was built to eat. Let's get into it. Here are 20 cancer-killing superfoods, ranked from remarkable to absolutely extraordinary. 1. Broccoli sprouts. Tiny food, massive power. Let's start with something that looks completely unimpressive. A small tangle of pale green sprouts you can grow on your kitchen counter for about 50 cents. Don't be fooled by the appearance. Broccoli sprouts are one of the most studied cancer-preventive foods in modern nutritional science. And the reason comes down to a single compound called sulforaphane. Here's what sulforaphane does. When you chew a broccoli sprout, an enzyme called myrosinase is released from the plant cell walls and converts a precursor compound called glucoraphanin into sulforaphane. That sulforaphane then enters your bloodstream and does something remarkable. It activates a protein in your cells called NRF2, which is essentially the master switch for your body's internal antioxidant and detoxification systems. NRF2 activation triggers the production of phase 2 detoxification enzymes, compounds your liver uses to neutralize and eliminate carcinogens before they can damage DNA. The research here is not preliminary. A landmark study published in the Proceedings of the National Academy of Sciences found that sulforaphane could reduce aflatoxin DNA adducts, a biomarker for liver cancer risk, by 50% in a clinical trial. Researchers at Johns Hopkins, where much of this work originated, have called sulforaphane one of the most potent inducers of phase 2 enzymes ever identified in food. Here's the critical detail. Broccoli sprouts contain anywhere from 20 to 100 times more sulforaphane precursor than mature broccoli. The same compound, same mechanism, but concentrated into a tiny sprout form that most people have never even considered eating. Grow them yourself with a mason jar and seeds, or find them at health food stores. Add them raw to salads, sandwiches, and grain bowls. Heat degrades the enzyme activity, so eat them cold. This is one food where the dose really does make the medicine. 2. Watercress, the peppery protector. Watercress has a sharp, peppery bite that announces exactly what it is: a deeply bioactive plant that your body recognizes as medicine. It's a cruciferous green in the same botanical family as broccoli and cabbage, which means it contains glucosinolates, the class of sulfur-containing compounds that the cruciferous family is famous for in cancer research. But watercress brings something specific to the table. It's an unusually rich source of a glucosinolate called gluconestertian, which converts in the body to phenylethyl isothiocyanate, or PEITC. PEITC has been the subject of considerable cancer research, particularly around its ability to interfere with the activation of carcinogens, specifically the kind activated by smoking tobacco. Studies have found that PEITC can inhibit cytochrome P450 enzymes that convert certain compounds in cigarette smoke into their most DNA-damaging forms. A study published in Cancer Prevention Research found that daily watercress consumption reduced DNA damage in blood cells. The effect was measurable after just 8 weeks. Meanwhile, watercress is also extraordinarily high in vitamin K, lutein, and beta-carotene, compounds that independently support cellular health and reduce inflammatory load. Eat watercress raw to preserve its glucosinolate content. Add it to salads, pile it onto sandwiches, blend it into green smoothies. Its peppery flavor means a small amount goes a long way. It's one of the most nutrient-dense foods on Earth by calorie, and one of the most underrated cancer-protective greens in any kitchen. 3. Purple cabbage, the anthocyanin armor. When you see purple cabbage at the market, what you're looking at is a food so drenched in cancer-fighting pigments that researchers are literally reverse engineering its chemistry to design pharmaceutical compounds. That deep purple-red color comes from anthocyanins, the same class of polyphenol pigments found in blueberries and cherries. But purple cabbage delivers them in a form that's shelf-stable, affordable, and extraordinarily concentrated. Here's what anthocyanins do at the cellular level. They are potent antioxidants that neutralize free radicals, the reactive molecules that damage DNA and initiate the mutations that can lead to cancerous cell behavior. But beyond antioxidant activity, research shows that certain anthocyanins can modulate cell signaling pathways, specifically pathways that regulate whether a damaged or abnormal cell is allowed to survive or is directed into apoptosis, programmed cell death, before it can proliferate. Purple cabbage also carries the full cruciferous toolkit: glucosinolates, indole 3-carbinol, and sulforaphane precursors. Indole 3-carbinol, formed when cruciferous vegetables are chewed or fermented, has been specifically studied for its effects on hormone-dependent cancers, with research suggesting it can shift estrogen metabolism toward less proliferative pathways. A study in carcinogenesis showed that cruciferous vegetable intake was inversely associated with several cancer types across large population cohorts. Eat purple cabbage raw and shredded in slaws. Ferment it into sauerkraut, which both preserves and enhances its bioactive content. Braise it low and slow. However you eat it, this is one of the most visually dramatic and biologically powerful anti-cancer plants available to anyone, anywhere, at almost any budget. 4. Kale, the cruciferous cornerstone. Kale became a cultural phenomenon and then a punchline, but underneath the tote bag jokes and the overpriced smoothies is a genuinely extraordinary plant with one of the most comprehensive anti-cancer nutritional profiles in the vegetable kingdom. Kale is cruciferous, which gives it the full glucosinolate toolkit. It's extraordinarily high in fiber, which independently reduces cancer risk, particularly colorectal cancer, by feeding beneficial gut bacteria, reducing transit time, and diluting carcinogens in the colon. And it contains high levels of carotenoids, particularly beta-carotene and lutein, which reduce oxidative stress in tissue. But kale's most underappreciated contribution is its vitamin K content. A single cup of raw kale provides several times the recommended daily intake of vitamin K1. Emerging research suggests that vitamin K plays a role beyond blood clotting, specifically in regulating cell cycle control and inhibiting proliferation of cancer cells in cell culture models. The research is early, but consistently interesting. Kale also provides sulforaphane precursors, quercetin, a flavonoid with anti-inflammatory and potentially anti-tumor activity, and kaempferol, a compound studied for its ability to induce apoptosis in cancer cell lines. Massage raw kale with olive oil and acid to break down its tough fiber and improve palatability and absorption of fat-soluble nutrients. Roast it into chips. Sauté it with garlic. Blend it into smoothies. Whatever format gets more of it into your daily diet is the right one. 5. Garlic, the allium defender. No food list about cancer prevention would be honest without garlic at or near the top. The evidence base here is extraordinary in its consistency. The World Health Organization's review of garlic's health effects identified meaningful associations between regular garlic consumption and reduced risk of stomach, colon, esophageal, and pancreatic cancers. That's not a single study. That's a pattern across dozens of population studies and clinical trials conducted over decades. The mechanism centers on organosulfur compounds, allicin and its downstream metabolites, diallyl sulfide, diallyl disulfide, diallyl trisulfide, have been shown to inhibit the activation of carcinogens, stimulate the activity of detoxification enzymes, suppress the proliferation of cancer cell lines, and trigger apoptosis in tumor cells across multiple cancer types. These compounds don't act through a single target. They appear to act through several simultaneously, which may explain why the epidemiological signal is so consistent across different cancer sites. One important note on preparation. Allicin is formed when garlic is crushed or chopped and exposed to air. The enzyme allinase converts alliin into allicin. Heat immediately inactivates this enzyme. So the best practice is to crush your garlic, let it sit for 10 minutes to allow the conversion to complete, and then cook it or eat it raw. That 10-minute window dramatically increases the bioactive compound content of whatever garlic you use. Raw garlic in dressings and dips. Cooked garlic in everything else. With the pre-crush. This is one food where technique genuinely changes the nutritional outcome. 6. Onions. The overlooked allium ally. Garlic gets all the attention in the allium family, but onions deserve equal standing. They are one of the richest dietary sources of quercetin, a flavonoid that has been studied in cancer research for its remarkable range of biological activity. Quercetin inhibits the enzymes involved in carcinogen activation, interferes with signaling pathways that drive cancer cell proliferation, and appears to sensitize cancer cells to apoptotic signals, while leaving healthy cells unaffected. Population studies consistently show that high allium vegetable consumption, onions and garlic together, is associated with reduced risk of gastric cancer and colorectal cancer. A meta-analysis published in Gastroenterology found that the highest allium intake group had a significantly lower gastric cancer risk compared to the lowest intake group. Onions also contain organosulfur compounds similar to garlics, though in different configurations, and they are an excellent prebiotic. Their fructooligosaccharides feed lactobacillus and bifidobacterium species in the gut. Given the emerging research connecting gut microbiome diversity with immune system function and cancer surveillance, this prebiotic activity may be part of the mechanism connecting onion intake with reduced cancer risk. Red onions carry additional anthocyanin content. Eat them raw on salads and sandwiches. Caramelize slowly until their sugars concentrate, or fermented in pickles. Daily use matters here more than quantity. 7. Mushrooms. The immune commanders. Mushrooms occupy a completely different biological kingdom from every other food on this list, and that difference is precisely what makes them so interesting in cancer research. Mushrooms are fungi. Their cell walls are built from beta-glucans, complex polysaccharides that are structurally unlike anything found in plants. And those beta-glucans have a specific, documented relationship with the human immune system. Beta-glucans are recognized by receptors on immune cells, particularly natural killer cells, and macrophages, and act as biological activators. They essentially prime your immune system to be more vigilant and more responsive. Given that cancer cells continuously produce proteins that signal the immune system to stand down, anything that keeps immune surveillance elevated and active is a meaningful ally. Some mushroom-derived beta-glucan preparations, particularly from turkey tail mushrooms, Trametes versicolor, have completed human clinical trials. A Phase 1 FDA-approved trial found that turkey tail polysaccharide K improved immune function in breast cancer patients following standard treatment. Shiitake, maitake, oyster, and lion's mane mushrooms all contain meaningful beta-glucan concentrations alongside other bioactive compounds, including ergosterol, a vitamin D precursor, and arytadinine. Cook your mushrooms. Heat ruptures cell walls and dramatically increases bioavailability of their active compounds. Sauté them in butter or olive oil. Add them to soups and grain dishes. And consider that every culture with a documented tradition of mushroom use, Japanese, Chinese, Eastern European, developed that tradition for reasons that modern immunology is now explaining in molecular detail. 8. Tomatoes. The cooked cancer shield. We covered lycopene in the context of collagen, but its role in cancer research is where the evidence is arguably even more compelling. Lycopene is a carotenoid, a fat-soluble antioxidant that accumulates in specific tissues, particularly the prostate, skin, and liver. And what it does in those tissues is neutralize singlet oxygen and quench free radical chain reactions before they can damage DNA. The epidemiological evidence connecting lycopene intake and prostate cancer risk is among the most consistent in nutritional oncology. Multiple large prospective cohort studies have found inverse associations between tomato product consumption, particularly cooked tomato products, and prostate cancer incidence. A landmark study in the Journal of the National Cancer Institute found that men in the highest quintile of tomato sauce consumption had significantly lower prostate cancer risk than those in the lowest. Here's the critical practical detail that cannot be overstated. Lycopene's bioavailability from raw tomatoes is modest. When you cook tomatoes, when you make sauce, paste, or roasted tomatoes, the heat breaks down the plant cell matrix and isomerizes lycopene into a form that your digestive system absorbs far more efficiently. Add fat, olive oil, butter, coconut oil, and absorption increases dramatically again. Tomato sauce cooked in olive oil is not just a culinary tradition. It is one of the most bioavailable lycopene delivery mechanisms that exists. Eat it multiple times per week. The dose-response relationship in the research suggests that frequency matters. 9. Pomegranate, the ancient healing fruit. If you want to understand why pomegranates have been symbols of vitality and healing in cultures from ancient Persia to biblical Israel to classical Greece, you only need to look at the compound profile of the fruit. Pomegranates contain punicalagins, hydrolyzable tannins that are unique to pomegranates and are among the most potent antioxidant compounds identified in any food. When metabolized by gut bacteria, punicalagins are converted to a compound called urolithin A, which has demonstrated anti-inflammatory, anti-proliferative, and immune-modulating effects in research. Pomegranate juice and extract have been studied extensively in prostate cancer research, where the results have been genuinely striking. A clinical trial published in Clinical Cancer Research found that daily pomegranate juice consumption significantly slowed the rise of PSA levels in men who had undergone surgery for prostate cancer, suggesting that pomegranate compounds were interfering with residual cancer cell proliferation. The PSA doubling time increased by 82% in the treatment group. That's a meaningful clinical outcome. Beyond prostate cancer, pomegranate extracts have shown anti-proliferative effects in breast cancer, colon cancer, and lung cancer cell lines. The compound ellagic acid, present in pomegranates, has been shown to inhibit tumor growth and sensitize cancer cells to treatment. Traditional Ayurvedic, Persian, and Mediterranean healers used pomegranate specifically for its healing properties long before any of this mechanism was understood. Eat the arils fresh. Drink pure pomegranate juice without added sugar. Add pomegranate seeds to salads, yogurt, and grain bowls. This is one fruit where the research fully validates the ancient reverence. 10. Blueberries, the antioxidant legend. Blueberries are perhaps the most studied berry in nutritional science, and their reputation in the cancer research literature is earned. They are extraordinarily rich in anthocyanins, the same class of pigments we discussed in purple cabbage, but blueberries deliver them in a particularly bioavailable form, alongside a complementary array of pterostilbene, resveratrol, and chlorogenic acid. The research on blueberries and cancer covers multiple fronts. Their anthocyanins modulate NF-κB, a master regulator of inflammation and immune response, and a pathway that is consistently overactivated in cancer tissue. Pterostilbene, a compound closely related to resveratrol, has shown the ability to induce cell cycle arrest and apoptosis in cancer cell lines while exhibiting significantly better metabolic stability and bioavailability than resveratrol itself. Research published in the Journal of Agricultural and Food Chemistry demonstrated anti-proliferative effects of blueberry extracts on breast cancer, cervical cancer, and colon cancer cells. If you're not already eating a large handful of blueberries every day, start today. Fresh, frozen, or freeze-dried, all forms retain meaningful polyphenol content. Combine them with a fat source to improve the absorption of fat-soluble antioxidants. Add them to yogurt, oatmeal, or smoothies. And consider that the populations in the United States, with among the highest wild blueberry consumption, traditional indigenous communities of the Northeast and Northern Canada, consumed them not as a trendy superfood, but as a staple, understood for generations to sustain health and vitality. I want to stop here for a moment. If what you're learning today is reshaping the way you think about what belongs on your plate, smash that like button right now. These foods are accessible to anyone. The science behind them is real. And every time you share this information, someone else gets to benefit from it. Hit subscribe and ring the notification bell so you never miss a video. We go this deep every single week. Now let's keep going. 11. Black raspberries, the overlooked cancer research berry. This is the one most people have never heard of, and it may be the most specifically and consistently studied berry in cancer prevention research. Black raspberries are not blackberries. They're a distinct fruit with an exceptionally high concentration of anthocyanins, ellagic acid, and ferulic acid, a combination that has attracted sustained attention from oncology researchers, particularly in the field of esophageal and oral cancer. The Ohio State University Comprehensive Cancer Center has conducted multiple clinical trials using black raspberry preparations in patients with precancerous lesions of the esophagus and colon. In one study published in Cancer Prevention Research, participants who consumed black raspberry powder showed significant decreases in biomarkers of tumor cell proliferation and significant increases in biomarkers of apoptosis in precancerous tissue. Those are not surrogate endpoints. Those are direct measures of biological activity in human tissue at the site of precancerous change. Black raspberries appear to work through multiple mechanisms simultaneously: antioxidant activity, anti-inflammatory effects, modulation of detoxification pathways, and direct effects on gene expression in tissue. The compound ellagic acid specifically has been studied for its ability to form adducts with carcinogens, essentially binding to them and preventing them from reaching DNA. Find black raspberry powder at health food stores and online. Add it to smoothies, yogurt, and oatmeal. Fresh black raspberries are seasonal, but worth seeking out. This is one of those cases where a lesser-known food has a stronger research profile than its more famous cousins. 12. Cranberries, the proanthocyanidin powerhouse. Cranberries are most famous for urinary tract health, but the compounds responsible for that effect, proanthocyanidins, also called condensed tannins, are generating serious interest in cancer research. Proanthocyanidins are large, tightly bound polyphenol chains that resist rapid digestion. They persist through the digestive system in forms that interact directly with tissue along the entire gastrointestinal tract. Research on cranberry proanthocyanidins shows anti-adhesion activity, anti-proliferative effects across multiple cancer cell lines, and the ability to modulate signaling pathways involved in tumor cell invasion and metastasis. A study in the Journal of Nutrition found that cranberry extract inhibited tumor cell proliferation and induced apoptosis in colon, prostate, and breast cancer cell lines. The effect was more pronounced than standard reference compounds in several cell models. Cranberries also contain ursolic acid, a triterpenoid with emerging anti-tumor research, and significant vitamin C content. The challenge with cranberries is that most commercial cranberry products are loaded with added sugar that counteracts many of the metabolic benefits. Seek out unsweetened cranberry juice, freeze-dried cranberry powder, or whole fresh or frozen cranberries, and use them in ways that don't require heavy sweetening. Their tartness is the sign of their potency. Embrace it. 13. Turmeric, the golden standard. Turmeric is probably the most widely discussed spice in the functional nutrition world, and for once, the hype is actually grounded in a substantial body of evidence. The active compound curcumin has been studied in over 3,000 published studies and has demonstrated effects across virtually every hallmark of cancer biology: tumor cell proliferation, apoptosis resistance, angiogenesis, the growth of new blood vessels that tumors require, invasion, and metastasis. The mechanisms are multiple. Curcumin modulates NF-kappa B, the inflammatory master regulator. It inhibits several kinases involved in cancer cell survival signaling. It sensitizes tumor cells to apoptotic signals, and it has been shown to inhibit the formation of carcinogens in the digestive tract. Research in human clinical trials has demonstrated that curcumin can suppress biomarkers of colon inflammation and precancerous lesion progression. Here is the critical practical information. Curcumin is notoriously poorly absorbed on its own. Two cofactors dramatically change that. Piperine, the active compound in black pepper, increases curcumin absorption by up to 2,000% by inhibiting its rapid metabolic breakdown. Fat further increases absorption of this fat-soluble compound. Golden milk, turmeric roasted vegetables, turmeric spiced lentils with black pepper and olive oil. These traditional preparations instinctively combined turmeric with both fat and pepper, for reasons that modern pharmacokinetics now fully explains. Never use turmeric without black pepper. Never eat it without fat. Those two conditions transform a spice with modest absorption into one of the most bioavailable anti-inflammatory and potentially anti-cancer compounds available from food. 14. Ginger, the ancient anti-inflammatory. Ginger has been used medicinally in Ayurvedic medicine, traditional Chinese medicine, and across cultures from West Africa to Southeast Asia for over 5,000 years. The modern science on ginger in cancer research is catching up to what traditional healers documented empirically across millennia. The primary bioactive compounds in ginger, gingerols in fresh root and shogiols in dried ginger, have demonstrated anti-proliferative and pro-apoptotic effects in cancer cell lines covering colorectal, gastric, ovarian, and pancreatic cancers. The mechanism with ginger is heavily anti-inflammatory. Gingerols inhibit the COX-2 enzyme, the same target as many pharmaceutical anti-inflammatory drugs, and suppress the production of inflammatory prostaglandins and cytokines. Chronic inflammation is now understood to be a foundational driver of cancer development, and ginger's ability to reduce that inflammatory load without the gastric side effects of pharmaceutical COX-2 inhibitors makes it a remarkable everyday tool. A study published in Cancer Prevention Research found that daily ginger supplementation significantly reduced biomarkers of colon inflammation in healthy volunteers at elevated colon cancer risk. The effect was measurable in tissue biopsies. Grate fresh ginger into teas, dressings, stir-fries, and smoothies. Use it liberally. It's one of the foods where the traditional wisdom that more is better actually tracks with what the research suggests about dose response. 15. Green tea, the EGCG elixir. If you had to choose a single beverage that has the most consistent research support for cancer prevention, green tea would be a very strong argument for the top position. The catechin EGCG, epigallocatechin 3-galate, is one of the most studied polyphenols in cancer research, and one of the most potent naturally occurring inhibitors of several kinases involved in cancer cell signaling. EGCG has demonstrated in vitro and in vivo effects across an extraordinary range of cancer types, and the epidemiological evidence from populations with high green tea consumption, particularly Japan, is compelling. Japan has among the highest per capita green tea consumption in the world, and among the lowest rates of several hormone-dependent and digestive cancers. Population studies from Japan have found inverse associations between green tea consumption and risk of certain cancers, particularly when consumption exceeds 3 to 5 cups per day. EGCG works through several mechanisms. It inhibits tumor cell proliferation. It suppresses angiogenesis, the growth of blood vessels that tumors hijack. It modulates apoptosis signaling, and it reduces systemic inflammatory burden. Research published in the Journal of the National Cancer Institute found significant associations between green tea consumption and reduced risk of certain cancers in prospective cohort data. Brew your green tea correctly. Water that is too hot, above about 80 degrees Celsius, degrades EGCG significantly. Steep loose-leaf green tea at lower temperatures for 3 minutes. Drink it without milk, which binds to catechins and reduces their bioavailability. 3 to 5 cups daily is what the research suggests for meaningful benefit. 16. Flax seeds, the lignin leaders. Flax seeds contain something that most people have never heard of, and that may be one of the most powerful hormone-modulating compounds available from food: lignans. Flax seeds are the single richest dietary source of plant lignans by a significant margin. They contain up to 800 times more lignans than most other plant foods. When gut bacteria metabolize these lignans, they produce enterolactone and enterodiol, compounds that act as weak phytoestrogens, capable of binding to estrogen receptors with far less potency than endogenous estrogen, effectively competing with estrogen at the receptor level and potentially reducing estrogen-driven cellular proliferation in hormone-sensitive tissues. The clinical evidence in breast cancer research is notable. Multiple observational studies have found inverse associations between high lignin intake and breast cancer risk, particularly in postmenopausal women. A study published in Clinical Cancer Research found that flaxseed consumption for a short period prior to breast cancer surgery resulted in measurable reductions in tumor cell proliferation markers and increases in apoptosis, markers in tumor tissue, a direct tissue-level outcome. Flax seeds also provide alpha-linolenic acid, a plant omega-3 with anti-inflammatory activity, and exceptional fiber content that feeds microbiome populations, critical for immune surveillance. Always use ground flax seeds, not whole. Whole flax seeds pass through the digestive tract largely intact. Grind them fresh or buy pre-ground and store refrigerated to prevent oxidation. Add 2 tablespoons daily to smoothies, yogurt, oatmeal, or baked goods. 17. Chia seeds. The fiber foundation. Chia seeds are extraordinarily high in fiber. A single 2 tablespoon serving provides nearly 10 grams, a remarkable density in a tiny package. And the research connecting dietary fiber with cancer risk reduction, particularly colorectal cancer, is some of the most consistent in nutritional epidemiology. The World Cancer Research Fund's Continuous Update Project, the largest systematic review of the diet-cancer relationship ever conducted, concluded that dietary fiber consumption is strongly associated with reduced risk of colorectal cancer. The dose-response relationship is linear: more fiber, lower risk. The mechanisms are multiple. Fiber reduces transit time in the colon, limiting the time carcinogens spend in contact with the mucosal lining. It dilutes carcinogenic compounds. It ferments into short-chain fatty acids, butyrate in particular, that serve as the primary fuel for colonocytes, the cells lining the colon, and that regulate their gene expression in ways that suppress cancer-promoting pathways. Research on butyrate in colorectal cancer prevention is extensive and consistently shows protective effects. Chia seeds also contain alpha-linolenic acid, quercetin, and kaempferol, overlapping anti-inflammatory and antioxidant compounds that complement their fiber activity. Soak them in water or plant milk overnight to form a gel that improves digestibility. Add them to smoothies, puddings, and baked goods. They are tasteless enough to add to virtually anything, which makes consistent daily use easy. 18. Lentils. The fiber and folate foundation. Lentils are one of the oldest cultivated foods on Earth, consumed continuously for over 9,000 years, from the Fertile Crescent across the Mediterranean and into South Asia. Every great traditional food culture developed legume-centered cuisine, not because plants were all that was available, but because experience taught them that these foods sustained health in ways nothing else replicated. Modern cancer research is now mapping out exactly why. Lentils are extraordinarily high in fiber, putting them among the top plant food sources. They're also one of the richest dietary sources of folate, a B vitamin that plays a critical role in DNA synthesis and repair. Folate deficiency impairs the cellular machinery that identifies and corrects DNA replication errors, errors that can initiate cancerous mutations. Adequate folate intake supports DNA methylation patterns that regulate the expression of tumor suppressor genes. The research on folate and colorectal cancer is among the most extensively studied diet-cancer relationships in oncology, with consistent protective associations. Lentils are also rich in resistant starch, a prebiotic fiber that produces particularly large amounts of butyrate during fermentation. Research from the World Cancer Research Fund identifies fiber-rich foods as strongly protective against bowel cancer with a grade 1 evidence level, the highest standard in nutritional epidemiology. Black, green, red, or French lentils. All varieties deliver meaningful fiber, folate, plant protein, and polyphenol content. A bowl of lentil soup several times per week is one of the simplest, most affordable, most evidence-backed dietary choices you can make for long-term cancer prevention. 19. Walnuts. The polyphenol powerhouse. Every nut has something to recommend it, but walnuts occupy a uniquely well-studied position in the cancer research literature. And the reason is their exceptional polyphenol content combined with the highest alpha-linolenic acid content of any common nut. Walnuts contain L-gitannins, complex polyphenols that gut bacteria convert to urolithins, the same class of compounds we discussed with pomegranates. They're also rich in juglone, a compound with demonstrated anti-proliferative activity in cancer cell models. Animal and cell research on walnuts has been strikingly consistent, with studies at UC Davis and other institutions showing that walnut consumption slows tumor growth across multiple cancer types. The Plant Foods for Human Nutrition Journal published research showing that walnut-enriched diets reduced markers of oxidative stress and inflammation in human subjects. A prospective cohort study found that nut consumption was associated with reduced all-cause mortality, with walnut consumption specifically showing associations with reduced cancer mortality. Walnuts' alpha-linolenic acid reduces the inflammatory prostaglandins derived from the omega-6 to omega-3 imbalance that characterizes most Western diets. Chronic inflammation, driven substantially by this ratio imbalance, is now recognized as a promoter of tumor development and progression. A small handful of walnuts daily, roughly 1 ounce, is what the research suggests for meaningful benefit. Eat them raw, not roasted, to preserve their delicate polyunsaturated fat content. Add them to salads, grain bowls, oatmeal, and yogurt. Their slightly bitter, earthy flavor is the signature of their polyphenol richness. 20. Seaweed, the ocean's cancer secret. And here we are, number 20. The one I saved for last, because it represents something no land plant on Earth can offer, and because cancer researchers have been quietly studying it for decades. Seaweed, specifically brown seaweed varieties like wakame, kombu, and bladderwrack, contains a compound called fucoidan. And fucoidan is one of the most biochemically unique compounds in any food on this planet. Fucoidan is a sulfated polysaccharide. There is nothing structurally like it in the terrestrial plant kingdom. It is built only by marine organisms, and it appears to interact with the human body in ways that are generating serious oncological interest. Laboratory research shows that fucoidan can induce apoptosis in cancer cell lines, inhibit angiogenesis, modulate immune activation in ways that enhance tumor surveillance, and in some models, inhibit the adhesion and migration of cancer cells. The cellular behaviors involved in metastasis. A study published in Marine Drugs found that fucoidan preparations extracted from brown seaweed inhibited tumor growth in animal models through multiple simultaneous mechanisms. Human clinical data is more limited, but the mechanistic research has been consistent enough that fucoidan supplements have entered clinical use in some integrative oncology practices in Japan and South Korea, where brown seaweed consumption has been a daily dietary staple for thousands of years. Seaweed also provides iodine in quantities found in virtually no land food, supporting thyroid function, which regulates cellular metabolism and immune activity. Nori, wakame, and kelp all provide meaningful fucoidan content. Add dried seaweed to soups and broths. Eat nori sheets as a snack. Use wakame in miso soup. The Asian longevity populations who eat seaweed daily didn't know about fucoidan, but they knew what this food did for their vitality over generations. Modern science is now reading the molecular fine print of that ancient empirical wisdom. If you made it all the way here, you've just received a complete, science-backed framework for building a diet that works with your body's own defenses rather than waiting for those defenses to fail. Let me leave you with the framework. Activate your body's detoxification and cancer surveillance systems with broccoli sprouts, watercress, purple cabbage, kale, and mushrooms. Foods that signal your cells to protect themselves. Add the allium defenders, garlic, and onions daily in forms that preserve their bioactive sulfur compounds. Flood your body with the polyphenol army from berries, blueberries, black raspberries, and cranberries, alongside pomegranate and walnuts. Use your kitchen spice rack as a medicine cabinet with turmeric, black pepper, and ginger combined correctly. Protect your gut, your first line of immune defense, with the fiber depth of lentils, flax seeds, and chia seeds. Target your inflammatory burden with green tea daily. Use cooked tomatoes to build lycopene protection in vulnerable tissue, and add seaweed to access the ocean's entirely unique biology. This is not a supplement protocol. This is a food strategy, and it's available to every person, at every age, starting with the next meal they eat. Smash that like button if this video changed the way you think about what belongs on your plate. Drop a comment and tell me which food surprised you most. Are you team broccoli sprouts, team black raspberries, or team fucoidan and seaweed? I read every comment. And if you're not subscribed yet, click that button and ring the bell. We're building a community that takes what goes on the plate seriously. Because the most powerful thing you can do for your long-term health isn't in a hospital. It's in your kitchen, and now you know exactly what to do with it.